Good day, everyone. My name is Ismail Ubaida Temitope, and I'm excited to share our findings with you today at the virtual conference on chemistry and its application, titled In Silico Study, focus on exploring the potential of phytochemicals from Vernonia amygdalina, commonly known as bitter leaf, as inhibitor of human androgen receptor and human glutamate carboxypeptidase 2. This research work was conducted at the Computational Biophysical Chemistry Unit of the Department of Pure and Applied Chemistry at Ladoke Akintola University of Technology in Nigeria. Here is my outline. Abstract. In this study, we investigate Vernonia amygdalina, commonly known as bitter leaf, known for its micronutrient content, as a potential treatment for prostate cancer using computational drug design method. A total of 167 previously identified bitter leaf phytochemicals, along with two standard drugs, apalitamide and hexalitamide, were subjected to evaluation for their inhibitory effect on the androgen receptor with the PDB ID of 1XOW and glutamate carboxypeptidase 2 with the PDB ID of 2XEI receptor utilizing computer-aided drug design methodology. Our, our result identified the listed compound as potential inhibitor of the two protein. This compound demonstrated favorable binding energy, drug likeness property, oral bioavailability, and bioactivity profile, suggesting their potential for further development as prostate cancer therapy. Today, I will be discussing a critical health concern affecting millions worldwide, cancer, particularly focusing on prostate cancer. As we all know, cancer is as a result of uncontrollable and unfavorable cell division, and this cell continue to grow, divide, and multiply into more dangerous cells throughout lifetime. There are many forms of cancer, which include breast cancer, lung cancer, leukemia, skin cancer, colorectal cancer, kidney cancer, and prostate cancer. Prostate cancer is one of the leading causes of cancer-related death among men globally, necessitating continuous exploration for new treatment avenues. This presentation aims to dive into the challenges posed by prostate cancer and explore innovative approaches toward combating these diseases. Stages of prostate cancer. Prostate cancer typically progresses through several stages, which are categorized based on the extent of the disease spread. We have stage 1 to stage 4. In stage 1, the cancer is still confirmed to the prostate gland and is often small and slow growing, usually not causing any symptoms. Also in stage 2, the cancer still remains within the prostate but may be larger than in stage 1. It may still be slow growing and might not cause any symptoms. But in stage 3, the cancer will have spread beyond the prostate capsule, but is still localized to nearby tissues, such as seminal vesicle. Symptoms may become noticeable at this stage. And in stage 4, the cancer will have spread to distant part of the body, such as bones or other organs. At this advanced stage, symptoms can be more severe and the cancer is harder to treat effectively. The staging helps guide treatment decisions and provide insight into prognosis of prostate cancer patients. Drug targets in prostate cancer. In prostate cancer, drug targets are specific molecules or pathways within prostate cells that are crucial for cancer growth and survival. These targets are these targets can include androgen receptor, which are central to the growth of prostate cancer cells through hydrogen signaling. Other targets may involve cellular processes like cell cycle regulation, DNA repair mechanism, and angiogenesis pathway, all of which play roles in cancer progression. Effective drug design to target these specific molecules or pathways aim to inhibit cancer cell growth, induce cell death, and disrupt tumor progression, potentially improving patient outcome and survival rates. Chemotherapy of prostate cancer and their side effects. Chemotherapy for prostate cancer involves using drugs to kill or slow the growth of cancer cells. Commonly used drugs like apalitamide and enzalutamide, which act as androgen receptor inhibitors, help to block the effect of androgen that promote cancer growth. However, this treatment can have side effects which include health problem and heart disease, erectile dysfunction, loss of sexual desire, cognitive dysfunction and memory loss. Therefore, there is need for other therapeutic agents. Phytochemicals and cancer therapy. Phytochemicals from various medicinal plants have shown excellent anti-cancer effects against different kinds of cancer with little or no side effects. And reports from experiments revealed that phytochemicals with antioxidative and anti-inflammatory properties can inhibit the growth of cancerous cells. So therefore, as all known, the Vernonia amygdalina, commonly known as bitter leaf, is a medicinal plant and it contains numerous micronutrients that are beneficial for human health and illness prevention. This bitter leaf contains phytochemicals such as flavonoid, alkaloid, phenolic, terpenoid, and so on. So 167 isolated phytochemicals from this plant were tested against two prominent proteins, that is the human androgen receptor and prostate-specific membrane antigen using computer-aided drug design. Computer-aided drug design is a computational approach used to identify and optimize drug candidates by stimulating their interaction with biological targets. We have like two type, structural-based drug design and ligand-based drug design. For this project, we employed structural-based drug design approach, which involve the identifying a target receptor, conducting virtual screening of potential ligands, that is our phytochemicals, and optimizing lead compounds based on their binding affinity and pharmacokinetic property. This process is faster, more cost effective, and has a higher success rate compared to traditional drug discovery method. Aim of the research is to investigate the anti-prostate cancer activity of isolated phytochemicals from Vernonia amygdalina, commonly known as bitter leaf, using computer-aided drug design approach (CAD). Materials and method. We started by identifying and preparing the phytochemicals and target receptor. The study utilized 167 phytochemicals from Vernonia amygdalina with the two standard, which is which are enzalutamide and apalutamide. The 2D structure of these phytochemicals were converted to 3D structure using Spartan 14 and optimized with DFT for molecular docking with the human androgen receptor and glutamate carboxypeptidase 2. The active side of these receptors were identified and validated using CASP and Behavior Discovery Studio software, ensuring accuracy against published experimental data. After which we checked the ADMET profiling and drug likeness. The ADMET profiling of the ligand were evaluated using ADMET SAS2 database, while drug likeness were assessed based on Lipinski rules of 5 using the Mole Inspiration online tools. These tools provide insight into the absorption, distribution, metabolism, excretion, toxicity, and overall drug likeness of the ligand. After which we carry out the molecular docking protocol. The molecular docking and scoring of optimized ligand and standard drug against the human hydrogen receptor with the PDB-ID of 1XOW and prostate-specific membrane antigen with the PDB-ID of 2XOEI were performed using Pyrex software. The inhibitor constant, that is the Ki, were calculated from the binding affinity, that is the delta G, in kilocal per mole using the equation Ki equal to exponential minus delta G over RT, demonstrating the ligand's potency against the target receptor. After getting our ligand with higher binding affinity, we check their oral bioavailability. Oral bioavailability assessment of the ligand were conducted using Swiss ADME web server. These tools provide insight into the potential oral bioavailability of the ligand, after which we check for their bioactivity test and pass analysis. Evaluating bioactivity properties such as ligand efficacy, fit quality, and ligand efficiency-dependent lipophilicity is crucial in early-stage drug discovery, as the metrics assess the potency, binding fit, and optimal lipophilicity of each compound, respectively, to ensure effective and safe drug candidates. Also, the biological activity of the ligand and the standard drug used in the research study were carried out using a web server. We check for the binding mode and molecular interaction, because identifying the binding site and evaluating ligand interaction in target receptors active pocket are also crucial in drug design, facilitating the improvement of ligand affinity during the lead optimization stage of drug discovery, which was using Discovery Studio and Pimo software. Research and discussion: structural elucidation and active site analysis of the two target receptor. The crystal structure of the human androgen receptor, the PDBID of 1SOW ligand binding domain bond with an NH2 terminal peptide, has been solved at 1.80 Armstrong resolution, revealing a monomeric structure with 12 alpha helix and 4 small beta strands, essential for male cells development and prostate cancer cell survival. The human glutamate carboxy peptidase 2 with a PDB-ID of 2xEI complex with antibody molecule has been solved at 1.69 Armstrong resolution, demonstrating its roles in enhancing immune recognition of prostate cancer cells by binding to prostate-specific membrane antigen and anti-DNP antibodies. Both structures provide critical insight into their respective active sites and contribute to the development of therapeutic strategy for prostate cancer. These are the amino acids present in the active site of the two target receptors. In the early stage of drug development, the advent profile, that is the absorption, distribution, metabolism, excretion, and toxicity of compounds, is crucial for selecting molecules with the best safety profile, as assessed using the advanced SARS-2 database. Out of 167 isolated compounds from Vernonia amygdalina, only 48 passed the ADMET screening, showing good human intestinal absorption, blood-brain barrier permeability, and non-toxicity, making them potential drug candidates. Among these, the ADMET result of the best 7 were displayed in this slide, whereas the standard drug was highly toxic, indicating its unsuitability as a therapeutic agent. Drug likeness analysis, which is based on Limpinski rule of 5. The rule of 5 in drug discovery stated that an effective oral drug should have a molecular weight no more than 500, hydrogen bond donor no more than 5, hydrogen bond acceptor no more than 10, and a log P not more than 5 to ensure good absorption and permeability. In the drug likeness analysis of the selected phytochemicals, 46 out of 48 ligands comply with the rule of 5, each having a no, each having no more than one violation. The screening confirmed the potential of selected compound and the best 7 results were displayed. Molecular docking analysis. The traditional approach to drug discovery is challenging due to its high cost, lengthy process, and low success rate, leading to the adoption of computer-aided drug design for its efficient speed and cost effectiveness. Molecular docking is a structural-based drug design method evaluate binding interaction between ligands and target receptor, with the rule that higher binding affinity results in lower inhibition constant. In this study, ligands that passed the ADMET and drug likeness screening were docked against human androgen receptor and glutamate carboxypeptidase 2, revealing binding affinity ranging from -8.7 to -7.6 kcal per mole for 1XOW and -10.4 to -8.4 kcal per mole for 2XEI, showing the 2XEI and the one with the highest binding affinity were displayed. Bioactivity analysis. The table shows the bioactivity characteristics of the selected heat and standard and the recommended value for bioactivity parameters such as ligand efficacy, fit quality, and ligand efficient dependent lipophilicity (LELP) are greater than or equal to 0.3, greater than or equal to 0.8, and -10 to 10 respectively. Notably, for the first receptor, the display compound has fulfilled all the requirement parameters and they were subjected to other analysis, which is oral bioavailability. Oral bioavailability is crucial for developing bioactive compound as therapeutic agent because low oral bioavailability can lead to unpredictable drug exposure. The Swiss ADME Web 2 was used to evaluate the oral bioavailability and physicochemical characteristics of selected compound and standard using the bioavailability radar to assess lipophilicity, molecular weight, polarity, solubility, saturation, and flexibility within recommended range. Past analysis. The past analysis predicted that all its compounds and standard have excellent anti-neoplastic activity with PA value greater than PI, indicating their potential for the design and development of new drugs for prostate cancer management. Binding mode and molecular interaction for the first receptor with a PDB-ID of 1XOW. Because determining the binding site and evaluating ligand interaction with the target receptor's active pocket are crucial in drug design, improving ligand affinity during lead optimization. For the androgen receptor, these are the compounds that bind with the active site, making them a potential inhibitor of the receptor, while the next slide showing the compound that bind perfectly with the active site of 2XEI, making them a potential inhibitor of the receptor with a strong interaction with their active site, suggesting their potential as effective inhibitor and than the standard. Conclusion. This study evaluates 167 isolated phytochemicals from Vernonia amygdalina plant for their inhibitory progress against prostate cancer receptor, that is the human androgen receptor and human glutamate carboxypeptidase 2, using a computer-aided drug design approach, which involves use of ADME-SAS2, mole inspiration web server, Pyrex docking tools, Swiss ADME, PASS software, among others. The results obtained revealed that the listed compound revealed them as the potential inhibitor of the two target receptor when compared with the standard, that is the apalutamide and enzalutamide, due to their good binding affinity, oral bioavailability, drug likeness, ADMET property, pass property, bioactivity, binding mechanism, and interaction with the active site of the target receptor. Lastly, apalutamide and enzalutamide, a currently used prostate cancer therapy, was found to have higher was found was found to have very large molecular weight, carcinogenic, hepatotoxic, inhibit some cytochromic P450. Therefore, drug administrators should reevaluate the potency as well as the toxicity of the drug in order to have a healthy life. Here are some of my references. All the authors are acknowledged and all the members of Computational and Biophysical Chemistry Research Group under the mentorship of Professor Abdulhamid Bilbaldin of the Department of Pure and Applied Chemistry, Ladoke Akintola University of Technology. Thank you for listening.